Hello, good morning, students. We have been discussing with regarding to Human Genome Project. We are talking about the methodologies of making human genome map. So there was two methods, sequencing, and second one was generation of physical and genetic map. We have spoken about sequencing, that is how to make the genome map by trying to arrange the sequence of nucleotide. There are two methods. One is expressing the nitrogenous base of what can say which are known to express as mRNA, that's only coding regions, and other one is sequence annotation method where both coding and non-coding regions are expressed. So to do this, we have got methods like isolate DNA, fragment DNA, separate DNA by gel electrophoresis, then try to join the fragments according to the what we can call sequence, that is based on the palindromic sequence using restriction endonucleases. So that's how we are going to break and join DNA fragment to get the entire length of DNA. So now in today's class we are going to discuss about the second step of methodology, generation of physical and genetic maps. So here the physical and genetic maps were generated by using information on the two important things. One is polymorphism of restriction endonuclease recognition site, and second one is some repetitive DNA sequences called microsatellites. That is the joint fragments of DNA. They should be arranged in a particular manner. Which segment will come after which? For that, that's what I told you. Restriction endonuclease enzyme recognition sites. So when you cut, the DNA will be cut only at that particular region where you will get what we can call cut fragments, which can be joined with complementary base pair of the other cut fragment. So therefore, that is done. That is the region where restriction endonuclease enzyme has cut DNA during fragmentation. So those regions will be identified. And the second thing is some repetitive DNA sequences called microsatellite. That is in human genome. You'll be studying after this. In human genome, there are certain nitrogenous base sequences which are repeated at frequent intervals in small number. So they are called as microsatellite. So try to identify those regions and join them. So this is how the physical and what we can call genetic maps can be made. So now we are proceeding further. That is, after making the human genome map, what we came to know, because when we had started human genome project, there were different ideas about human genes. There are so many genes. They are made up of so many bases. They are going to express in particular pattern. All these things were known to us. Now the question is, all that what we had hypothesized or what we had believed earlier, is it the same? So after getting human genome project, whether all those characters are seen in this. So therefore, in salient features of HGP, we are going to study 10 important characteristic features which we have observed after making the map. So now let me come for one by one the salient features. The human genome contains 3164.7 million nucleotide bases. So earlier it was told 3 into 10 to the power 9 base pair. Yes, almost it comes to the same. So therefore the first concept or first belief what scientists had believed about human gene, particularly with regarding to number of nucleotides, is almost correct. So therefore human gene contains 3164.7 million nucleotide bases. Second thing is the average gene consists of 3000 bases, but sizes vary greatly, with largest known human gene being dystrophin with 2.4 million bases. If there was a question here, what's a gene? So what is the number of nitrogenous bases that represents one gene? So therefore, here the nitrogenous base sequences number varies from one gene to another. But one thing we have come to know after making Human Genome Project is the average gene is known to have 3000 bases. So that is the standard number of nitrogenous bases that represent one gene. But this keeps on varying as the Human Genome Project has highlighted us. And we have the largest known gene, and that largest known gene is dystrophin, and it is made up of 2.4 million bases. Remember, starting from 3000, we have got a gene containing 2.4 million nitrogenous bases. So this is the second important salient feature. Now, the third one, the total number of genes is estimated at 30,000, much lower than the previous estimate of 80,000 to 1,40,000 genes. That is, earlier scientists believed that human contains, or an human being is known to have at least 80,000 to 1,40,000 genes. This was our assumption before making human genome map. But after this, yes, we came to know that an human being, individual, is known to have 30,000 genes. So this is very, very low when compared to our earlier assumption. Then the fourth important characteristic feature is almost all nucleotide bases, that is 99.9% bases, are exactly same in all people. Yeah, this is very, very important. So we say human beings belong to one species. Why means this is the main important reason. That is, the genes, they contain same nitrogenous bases maximum, that is 99.9% genes are same in all people. This is the fourth important salient feature. The fifth one, the functions of over 50% of discovered genes are unknown. Yes, we have discovered 30,000 genes. Of that, we don't know the function of 50,000 genes. That's the thing. So why they are present? What is their function? That has to be yet discovered. So therefore, even after making human genome map, scientists know only about 50,000 genes and their function. Remaining 50,000 genes functions are not known. Then the next important thing what we know is less than 2% of genome codes for proteins. Yes, very, very important is this. So we have got 100% genes, but of that only 2% or even less than that are going to code for proteins. Yes, you'll be studying in molecular biology. That is, DNA prepares mRNA. mRNA carries message for proteins. Proteins are known to be a part of function or a part of structure. So therefore, to code for that protein, the genome required is less than 2%. Then what is the role of 98% of genome in human being? That is not known. So therefore, this is the other important salient features of Human Genome Project. So we will continue with remaining four salient features in the next slide.